Okay, grade 10, so part 3, but you know this one, okay? Cross-section of a leaf. I sit here and I look around me and I've got models and I've got books and I've got pictures and I've got crayons, Copies to color in, and I've got scanned pictures. You know, biology is interesting. Just remember that, okay? There's that whole world out there that you don't even know about, and I certainly don't know enough about everything that goes on. And I just hope that you have maybe seen the wonder of plants and not necessarily, oh no, we're doing plant issues, but that you can actually see that there's so much diversity there and it's also ordered and structured, just like our bodies. I mean, you learned something about the human body last year, the circulatory, the digestive systems, gaseous exchange. You've learned about the reproductive system. It's just amazing. And even though everyone's not such a keen botanist as myself, I just enjoy it. Okay. Um, just try and see the wonder in everything that we have been talking about. All right. So, like I said, part 3, let's have a look at the transverse section of the leaf. And this one you are familiar with, very familiar with. All right. So, what do we have here? Once again, that same old diagram, the transverse section of the leaf, which I showed you yesterday with this beautiful photograph. And there's the line diagram. And here is mine. Unfortunately, whoops, okay, I've been demoted. Okay. Unfortunately, I've gone down to the ground floor. Sorry. Okay. Unfortunately, the diagram, when I scanned it, it cut off a little bit here and it kept doing that. So we're just going to work with this one. All right. Now, something that I haven't mentioned here is that leaves are dorsiventral. That is a lovely word. It explains that in dicot leaves, so leaves of roses and apple trees and dandelions and things like that, not grasses. Grasses, their leaves are kind of the same on both sides because grass leaves stick up, or lily leaves and things like that, they're strap shaped, and what's on this side is on that side. Not much difference. But dicot leaves are dorsiventral. The top part you can mention, I mean, you can recognize, and the bottom part. And remember, every time I use that model, I said, okay, not this way, put it the right way around, because they're dorsiventral. So just like a stem and a root, the leaves are covered by an epidermis. And we know that the upper epidermis has fewer stomata. I haven't even drawn any in here. And there's a thick waxy cuticle secreted by those cells. So there's upper epidermis, thick waxy cuticle, and it goes all the way around and becomes over the whole bottom of the leaf. Remember, leaf, upper epidermis, lower epidermis, whole thing. Okay, so the lower epidermis, you can see I've drawn in some of the epidermal cells and also the guard cells, but I've colored those in green and I've left the little opening in between blank because that's where gaseous exchange is going to occur through the stomata. So 1, 2, 3, I've drawn 4 stomata, there's another 4, but it's just on that one, we're cutting across the leaf. There'll be so many. And you can see there's a thinner waxy cuticle secreted by the lower epidermis. And then I've drawn, once again, the bean-shaped guard cells moved apart to show the stoma. But when you're looking from the side, you're cutting across those cells, and that's why they look like little circles. You're not looking from the top anymore. All right, then how do we know which side of the leaf is which? Well, okay, so we've got to look for the veins. So in the middle, that middle vein of the leaf, and then you've got the branch veins that branch off the mid vein. I don't seem to have that model here now, but I'm sure you remember. Xylem is always at the top, okay? Phloem below. We have written it in, okay? So there's sieve tube cells, the little colored in ones I've made companion cells, and the sieve plate. Remember the sieve tube cells have got little perforated plates at the end that the sugars pass through. And then at the top are little strands of xylem made up of bigger vessels and smaller tracheas. And then that vein and the little side veins, all right, are always strengthened in some way. So I've only drawn sclerenchyma, but I've said here or parenchyma or both. And you saw that in the model. Okay, we've done this now. And then I forgot to write that on here. The mesophyll, MESOPHYLL, meso, middle, full leaf. There's the middle of the leaf. It's filled with photosynthetic tissue. There's palisade parenchyma or palisade mesophyll. Oblong cells closer together, not such big air spaces. And then spongy parenchyma below, much bigger air spaces connecting with the stomata so that it's easy for gaseous exchange to occur. All right, so let's just check if I've covered everything. Just read on the side. It's upper epidermis, palisade parenchyma, branch vein or vascular bundle, a little vascular bundle. Lower epidermis, stoma or pore, spongy parenchyma. There I've added in guard cells in sections. So, I mean, I don't expect you to do all of this, but now you are going to, once you finish watching this video, you are now going to go and fill everything in, and that becomes your summary notes. And phloem below. Okay. I think I've covered everything. Yes, I have. All right. So let's switch this one off, and you are now going to add to your diagrams for the remainder of this double lesson.